Thank you very much, and thank you for the organisers for the invitation to be here today and to share with you some exciting data from the lab that I work in and also the potential implications of this for the treatment of patients with cancer, especially in the early stage setting. So the premise of what I will talk about and the main background is that cancer is an evolutionary process. We know that from the moment we have a fertilized egg and the zygote, that throughout childhood and adulthood that we're exposed to various lifestyle, environmental exposures. Some may be known to be carcinogenic. These may be extrinsic factors, but also there are intrinsic factors. With every cellular division, there'll be a degree of accumulation of abnormal genetic events, some mostly relating to DNA repair, but that over childhood and adulthood in one's lifetime, one acquires these abnormal genomic events, either mutations, so point mutations involving abnormalities in the DNA code, but also on a larger scale involving chromosomes, so gains and losses of chromosomal arms, and I'll refer to that as copy number genomic abnormalities. And if one is then to go on and develop a tumour, a benign that then becomes malignant tumour, it therefore follows that given there is this evolutionary process that tends to follow, truly follows Darwinian natural selection, that you end up with a heterogeneous mass or tumour. And then if you then go on and offer these patients targeted therapies or chemotherapy, these are so-called selection pressures that may select for some populations of cancer cells as opposed to others, depending on sensitivity and resistance to drugs. And so you end up with an even more heterogeneous mass or tumour, depending on the treatment that you offer patients. So these tumours are composed of populations of cancer cells that all derive from one cancer cell, but that are heterogeneous in terms of genetics. So why might it be that when the patient with cancer presents to you, a single diagnostic biopsy does not quite capture the information that you need in order to offer them the treatment that they require? So one of the main interests in the lab that I work is intratumour heterogeneity. It's not a novel concept. We've known about it for more than a century. Pathologists would comment, looking down a microscope, that there's certainly evidence of differential protein expression. So we know that there's heterogeneity in terms of the phenotypic picture of tumours. But we now know, at least from the last 5 to 10 years, small retrospective genomic cohort studies that involve genetic sequencing, either the whole exome, the 1% of that codes, the coding part of your whole genome, or even whole genome sequencing, that tumours are heterogeneous. No two patients behave the same, even with the same tumour type and histological subtype. Their disease course varies from one patient to another. We know that on the level of the tumour, there's intratumour heterogeneity, so what we refer to as these subclones or populations of cancer cells, intratumour heterogeneity, and that's also the case on a cell-to-cell intercellular level. So again, these are tumours composed of different clones or subclones, populations of cancer cells that are genomically, genetically related. And we know this is the case in many solid tumour types, also hematological malignancies, and this is really just to depict that various studies have demonstrated this Darwinian natural selection and process of evolution from early stage to late stage disease. So I want to tell you a little bit about the lung tracer X study, and in fact we've looked at this in kidney cancer and melanoma. There are no studies that look prospectively to see what is the clinical relevance of intratumour heterogeneity or tumour evolution in terms of clinical outcome and therapeutics, drug response and resistance. So in this particular study of 800, 900 patients, they present with non-small cell lung cancer. They're eligible for curative surgical resection. They're recruited into the study. We follow them up to 10 years. Their cancer recurs, they develop metastatic disease, and there's a process of longitudinal sampling of tissue and blood up to the point of metastatic disease. And this is a way of trying to understand prospectively how early stage disease and metrics of intratumour heterogeneity correlate with clinical outcomes. And I'm going to share with you some of the data from the first 100 patients, and we've seen similar results in the following 200-300 patients. So far, we've recruited over 700 patients into this study that opened 5 years ago. So surgically resected samples, we look at multi-regional samples, whole exome, deep, that means deep coverage, level of sequencing. We look at mutations, but also chromosomal events or copy number events, and we're able to decipher so-called phylogenetic or evolutionary trees. A lot of the terminology comes from classical ecology. And we look at the presence and absence of genetic abnormalities throughout the tumour mass, and we look at the clones that harbour these genetic abnormalities. And on an evolutionary or phylogenetic tree, the trunk represents clonal mutations or genetic events. These are present throughout the tumour in all cancer cell, versus those on the branches or subclonal genetic events. The subclonal branch events occur later on in tumour evolution. As opposed to clonal, these are abnormalities present from the very beginning, that cancer cell that goes on to lead to a heterogeneous tumour mass. This is really just to visually depict. Red represents subclonal on the branches, blue represents clonal in the 100 patients we've looked at so far. Clearly, there's evidence of extensive genomic heterogeneity. There are many subclonal genetic events that occur later on in tumour evolution, both at the mutational level but also at a larger chromosomal event level. So that's fine. We, in keeping with other studies on a larger scale, demonstrate that tumours are heterogeneous, but what does this mean in terms of clinical outcome? How might we use it in order to better stratify our patient treatment and follow-up? So it turns out, even in the first 100 patients, that there is prognostic relevance of tumour heterogeneity. But surprisingly, not when you look at mutations, individual mutations, against which many drug studies have established targeted therapy as the so-called magic bullet. So if you look at the level of heterogeneity, those that are branch subclonal versus those that are clonal in patients' tumours, you find that there's no relevance in terms of clinical outcome and disease-free survival. However, when you look at copy number aberrations, which is an indicator of chromosomal instability, if you like, this is statistically significant in terms of prognosis. So if you have a tumour, a cancer that has a high degree of heterogeneous chromosomal events that have occurred later on in tumour evolution, you're more likely to experience disease relapse early on and the development of metastatic disease and poorer overall survival. So in the interest of time, I'm not going to go through the data that supports this, but we have data that demonstrates why this might be. We see that chromosomal instability drives parallel evolution. So these different populations of cancer cells have abnormal genetic events that occur involving the same gene, but in different alleles, paternal and maternal alleles. So there's a drive for the same signaling pathway that involves an oncogene that is predicted to drive a cancer occurring in both alleles that the patient would have inherited from mother and father. We also see that there's good evidence across the cohort, 40% at least, of HLA loss. So we know that cancer cells present antigens to the immune system, and that if there is a loss of HLA, we need HLA in the interaction with MHC in order for antigens to be recognized by the immune system. If there is a loss, then these tumours are able to evade the immune system. So there's good evidence of chromosomal instability driving immune evasion. And I will touch on this later, but we also see that in that primary tumour that was surgically, in theory, curatively resected, that one populational subclone of cells that harboured a high degree of chromosomal instability, it's that's the lethal clone that goes on to lead to metastatic disease. So chromosomal instability and poor outcome may be explained via various mechanisms. Just to pause, what does that mean in terms of therapeutics? So clearly, there's spatial heterogeneity in these tumours. So your single diagnostic biopsy not only does not capture the entire genomic landscape of the tumour, but it also might limit your ability to identify a predictive marker that might be relevant for therapeutics, for example. Also, if you've got a heterogeneous tumour mass, and there'll be more, say, clones of populations of cells that contain resistant mutations, this might be an ideal substrate for tumour evolution. So somehow we see prospectively and also in the retrospective studies that the more heterogeneous the tumour, more likely you are to develop resistance to drugs and therefore therapeutic failure. And also we don't know if you identify a druggable or targetable genetic event, mutation mostly, if it's on the branch, subclonal versus clonal on the trunk, does that make a difference in terms of their response to therapeutics, in terms of their progression-free survival? And in fact, off the back of this study, there are various drug studies that we've set up to try and establish whether targeting an event on the trunk versus the branch means you are more likely or less likely to respond to drugs. And it's disappointing that in the context of therapeutic drug development, very few studies, if any, take into account heterogeneity and tumour evolution. We often rely on an archived single diagnostic biopsy to make decisions in patients who years down the line present again in clinic with a disease relapse. We know that cannot be reliable given that these tumours evolve. So how might we leverage heterogeneity to our benefit therapeutically? If these tumours are heterogeneous and there's chromosomal chaos or instability, how might we take advantage of that? So one approach is to leverage two of the new hallmarks of cancer. So one is that we know these tumours are heterogeneous and have a high degree of genomic instability, and also that they are able to evade the immune system. And it seems logical to say that why not target clonal mutations, because they're on the trunk, they're present throughout the tumour in every cancer cell. And why not target multiple, given that that might give you the best chance of a therapeutic response? So I mentioned tumour antigens or neoantigens. These are specific to every cancer cell. You need HLA and MHC interaction in order for these to be recognised on the cell surface by CD8 T cells. When we look at the TRACERx cohort of 100 lung cancer patients, both in adenocarcinoma and squamous cell histological subtypes, we see that if you look at the total number of neoantigens, the clonal number of antigens and the subclonal, it's not surprising that it's the patients who have tumours with a high proportion of clonal neoantigens who fare better in terms of clinical outcome. And you might say that's because there's a high chance that there would be immune recognition. So this doesn't tell you much about therapeutics, but that patients who have clonal neoantigens high burden are more likely to do better in terms of clinical outcome and survival. And so we're about to set up next month a phase 1, phase 2 study trying to demonstrate that if from patients' tumours we can detect these neoantigens and one can predict them bioinformatically, if you design peptides that match these antigens, and peripheral blood allows you to extract antigen presentation cells as well as TILs ex vivo, you can expand these active T cells that recognise these neoantigens and reinfuse this back into the patient, so an adoptive autologous T cell therapy. And we'll see whether or not this matches the science and the basic science that we see in the lab. So I've talked about early stage, curatively resected tumours, but how do cancers evolve to then go on and develop recurrent and metastatic disease? After all, it's overwhelming metastatic disease burden that kills these patients ultimately and leads to multi-drug resistance. So the PEACE study is a national autopsy programme across the UK, and the aim is to recruit all patients with solid cancers and metastatic disease. And within 24-48 hours, we perform a targeted post-mortem or autopsy to collect tissue from all sites where the cancer is spread to try and understand better at the advanced stage how the metastatic process occurs and why patients develop drug resistance. And just a bit of, we've already recruited over 150 patients into that study, but just to give you two examples, this is a patient with lung cancer. We were able to collect tissue from the primary tumour. They were in the adjuvant setting, disease-free, and then developed disease recurrence. We collected tissue from the relapse biopsy, and then they go on to develop metastatic disease. And at autopsy, we're able to collect fresh tissue, and when you put it together, you come up with an evolutionary tree, which you see here, that tries to demonstrate tumour evolution from early stage to late stage disease. So these blue and green here represent two populations or subclones in the primary tumour that then go on to seed sites of metastases in the brain, in the adrenal gland, in the lung, in the liver. And it appears that there are certain genetic patterns, both in the chromosomes and mutations, that seem to predispose these clones to certain sites of metastases. So this is one way of being able to understand tumour evolution in the metastatic setting. And just to demonstrate, this is another patient with kidney cancer. When we look at the primary tumour subclones and metastases, we can identify that the lethal subclone that goes on to develop metastatic disease and seed cancer is the clone that harboured the highest degree of chromosomal instability. Again, we see this macro event of chromosomal instability playing a role in disease progression and poor outcome. What can we realistically do, though, to track tumour evolution. It's fine if it's a surgical specimen or an autopsy, but you can't offer patients 5, 10 diagnostic biopsies to try and capture spatial heterogeneity. So we know that liquid biopsies and circulating biomarkers are able to also track tumour evolution, and again in the first 100 patients in the lung study, and we've seen this again in kidney and melanoma, if you know some of the genetics of the primary tumour, you can design bespoke panels in the blood, and you can look at circulating tumour DNA and circulating tumour cells and actually map the phylogenetic evolutionary tree, but from the plasma. And this predicts the early emergence of resistance. You can predict the evidence of disease relapse up to 6 months before you radiologically or clinically see any evidence of disease recurrence. So this can at least be used in the setting of early stage disease or the ability to predict disease relapse. And we've opened again a phase 2 study where we're trying to see whether or not these circulating biomarkers can detect what's called minimal residual disease and see whether that can be used to stratify patient treatment and improve clinical outcomes. And if you're able to capture this early on in tumour evolution, before these patients have developed secondary or metastatic disease, you may be able to intervene and improve their survival. This is my final slide, so I hope I've convinced you that tumour evolution is real. I've focused on lung and kidney, but we've seen it throughout solid tumour types. And the future. Perhaps drug development strategies need to incorporate tumour evolution. We're certainly trying to look at that in lung cancer off the back of the TRACERx study. Could it be that the more lung cancers and other cancers that we study prospectively, we might be able to come up with so-called evolutionary rule books? If one can predict the oncogenic event that follows B, that follows A, one might be able to either offer patients combined therapies or predict the next evolutionary step and be one step ahead of the tumour. Could it be that circulating biomarkers will be our route to try and understand tumour evolution using non-invasive techniques? And actually, given that immune recognition, and we've seen early on with immunotherapy, that patients have remarkable treatment response when we're focusing on leveraging the immune system, perhaps leveraging clonal neoantigens and using intratumour heterogeneity might also lead to better outcomes. And I want to end on saying that I've focused heavily on genomics, but a cancer cell is malignant and goes on to develop true clinical tumours depending on its context, its tumour microenvironment. So we try to correlate the genetics with the immune microenvironment, vascular supply, hypoxia, and so on, but also to look at genetic expression, epigenetics, and the metabolic state of these tumours. Thank you for your attention.